welcome everybody to my webinar today. It's titled Rethinking the Traditional Western Blot. My name is John Lysate, and I'm a field application scientist with Core Biosciences. So for my talk today, I want to talk about the In-Cell Western Assay. I want to talk about what it is, its benefits, and how you can use it in the lab in your everyday experiments. I also want to talk about a variation of the In-Cell Western Assay, which we call an On-Cell Western Assay. After I go over the assays, I then want to talk about a case study in which a group uses the In-Cell Western Assay to study Alzheimer's. So what is the In-Cell Western Assay? The In-Cell Western Assay begins with plating your cells in a microtiter plate. These plates can range from anything from a 6-well plate up to a 1536-well plate. The one requirement for these plates is that they're clear bottomed for laser detection at a later step. Now once you've plated your cells, you wait until they're of confluency. Now these cells can be either adherent or suspension cells. Now once the cells have reached desired confluency, you then do your treatment of choice. And as you'll see later, this can be either adding a ligand, adding an inhibitor, siRNA incubations, whatever you're interested in. You incubate the cells, remove the media, and then fix and permealize those cells. Once you've permealized, you then incubate with antibodies. Primary antibodies specific for your target of interest, and then secondary antibodies which are coupled to fluorophores. In the case of the In-Cell Western, these fluorophores are coupled to near-infrared dyes called IR dyes. These dyes either fluoresce at 680 or 780 nanometers and allow for detection on an Odyssey CLX, Odyssey Classic, or an Odyssey SA. The Odyssey systems are point-based scanner systems that utilize these two near-infrared lasers, and because you have two lasers, you can simultaneously detect and quantify two separate proteins and signals at once. Now this gives you an idea over the general protocol, but let's talk about workflows and why this is something that would be beneficial for you in the lab. We'll compare the In-Cell Western Assay, or ICW, to traditional Western blots or immunoblotting. Typically right now, after you do typical cell incubations, you're lysing those cells, transferring those lysates to the lab where you're potentially spinning out insoluble materials and then fractionating, in some cases, your sample to obtain a pure sample. Once you have your final sample, you then run SDS-Page gels and then transfer the proteins to a membrane, PVDF, nitrocellulose, and begin your antibody incubations. Now, depending on length of time spent at each of these steps, the traditional Western blot can take over 6 hours, and in some cases, even longer. With traditional Western blot, some steps such as spinning out insoluble materials, transferring to membranes, can decrease sensitivity because sometimes your protein of interest may be lost at some of these stages. Now with the In-Cell Western Assay, after you're done with your cell treatments, again, you simply fix and then permealize the cells. Once you're done with this step, you move directly into antibody incubations, cutting out a lot of these middle steps involved with the traditional Western blot. Because you remove these costly time steps, the overall time spent with the In-Cell Western is much shorter than the traditional Western blot. Now, one key distinction between these two protocols is typically you're probably denaturing your samples for your Western blots. Since we're fixing and permealizing these cells directly in the well, In-Cell Western looks at the native state of the cell. Because of this, one consideration when adapting the In-Cell Western to the lab is that primary antibodies used should recognize native epitopes and not denatured epitopes. Now, in addition to the time saved with the workflow, you also will save time in just number of samples run and reagents run. Shown here is a 384-well plate And if you use a 384 well plate for an In-Cell Western, that's 384 different data points you can obtain at once. To obtain that same amount of data with traditional Western blots, you need to run roughly 43 10-well gels. So what this means for you is you get a lot of data quickly in a shorter period of time. Now, in addition to this time-saving component, the data you get is also of higher quality. Shown here is some data obtained in 2008 by a group that did the same experiment with their traditional Western blot protocol and then the In-Cell Western Assay. Now, the specifics of the experiment for this purpose aren't too important. The main thing to pay attention to is that they were screening 20 different mutants, and they were screening for these mutants for changes in protein phosphorylation. So when we look at these two graphs, the y-axes are plotting increases in protein phosphorylation over controls. Now, when comparing the In-Cell Western data to traditional Western blot data, the first thing to notice is that the trends are the same. So if you have a mutant that has a high level of phosphorylation, it's going to give roughly the same level of phosphorylation as read with an In-Cell Western. So higher levels here, and then with mutants with lower levels shown here, you can see the same trends with traditional Western blot or the In-Cell Western. The key difference in the data is when you compare error bars from sample to sample, Western blot error bars are much greater than corresponding In-Cell Western error bars. This is because since you remove a lot of the steps from the traditional Western blot, because you remove those steps in the In-Cell Western, you have higher reproducibility, replicate to replicate, experiment to experiment, giving you more significance in your results. Now, results shown here with asterisks are significant. So when they screened these mutants, they were only able to find 4 significant hits with traditional Western blots. Same experiment done with the In-Cell Western, you can now find 15 significant results that were significantly higher than controls. So the In-Cell Western will allow you to save time in the overall workflow. You get more data, and the data you obtain with the In-Cell Western will be of higher quality. So let's talk about a couple of examples of how you can use this in the lab in your everyday research. And let's start by talking about protein phosphorylation. Let's say you've seeded your cells of interest in a plate of choice, and that these cells express a receptor that your group researches. And let's look at a typical signal transduction pathway. And so when we have our cells at the desired confluency, we're going to go ahead and add a ligand for our receptor of choice. Once we've added ligand, we can incubate the cells and monitor downstream phosphorylation of our targets of interest. Now, since you're using microtiter plates, whether you like 96 or 384 well plates, from well to well you can vary very easily the amount of ligand you add, whether you add inhibitors, or the time period that you allow for incubation. Now, once you've incubated these plates with the desired ligand concentration for the desired period of time, and you have sufficient phosphorylation of your target, you can then go ahead, remove the media again, and fix and permealize those cells. Now, once those cells are permealized, you can add primary antibodies against your target of interest. So for this example, what we're trying to monitor are changes in protein phosphorylation. So our primary antibody is going to be specific for that target. After we've added primary, we add secondary antibodies, which again are coupled to IR dyes, which will allow for detection on an Odyssey system. Now, if you recall what I had stated previously, is that the Odyssey system utilizes 2 near-infrared lasers for simultaneous detection of 2 signals. So here, what we're doing is we're monitoring protein phosphorylation. But a key question is, how do we normalize data from well to well? Changes in cell density from well to well will throw off the accuracy of our data. And so by utilizing the Odyssey system, in one laser channel, we can monitor our protein of interest or phosphorylation of interest, and then in the second channel, we can have a normalizing signal. Typically, you might see normalization to things like a total protein signal. So if you're monitoring something like ERK and phospho-ERK, your normalizing signal would be your antibody directed against your total ERK signal. Conversely, you might also see primary antibodies directed against a housekeeping protein, such as actin, tubulin, or GAPDH. Now, the downside to using antibodies directed against either total or housekeeping proteins is that they require, one, you have the appropriate primary antibody. These primary antibodies have to be validated. And secondly, if your total and housekeeping protein are of the same species as your target of interest, it makes it much more difficult to segregate them into the two laser channels. So to eliminate these hurdles involved with using either total or housekeeping protein antibodies, many groups use nonspecific cell stains to normalize against cell number. Traditionally, you may have seen labs use either the DRAC5 stain or the Sapphire 700 stain. DRAC5 stain is a nonspecific DNA stain. Its linearity over cell density is plotted here below with squares. When you look at the linearity, you can see at lower cell densities, it has a decent linearity, but at higher cell densities, it plateaus off. Sapphire 700 stain, on the other hand, is a nonspecific nuclear and cytoplasmic stain. It's plotted here with circles, and its linearity is typically limited to higher cell densities. So the workarounds for using these two stains has been typically to combine them both. That when you combine them both, that's plotted here with triangles. And you can see when you combine both stains together, you have a linearity over the entire cell range. Now, some of the issues for you in the lab, if you use both these stains, is one, you're going to have to optimize both these stains separately for your cell of interest. These reagents are costly. And to eliminate some of these hurdles with using them, Lycor now offers a cell tag 700 stain. This is a single reagent that nonspecifically binds to both nuclear and cytoplasmic stain. And it works best over both low and high cell density. Now, once you've chosen your normalizing signal, this is what the typical data will look like. So this is an experiment in which EGF was added to cells. Shown here in the center in green, on the 800 channel of an Odyssey, is primary antibody directed against EGF receptor. So this is our normalizing signal. Shown below in red, on the 700 channel of an Odyssey, is our phospho-EGF receptor signal. Now, if we start on left, when you have no EGF receptor, you can see you have receptor expression shown in green, and then no low levels of phosphorylation, shown in red. And then in the top image is the overlay of the two. When you add EGF, you see high levels of phosphorylation. Now, what this data is showing is the addition of an inhibitor as well. Working from right to left, you have increasing concentrations of inhibitor. What you can see is at low levels of concentration of inhibitor, shown on right, you have high levels of receptor phosphorylation. As you increase inhibitor, you block that phosphorylation. Now, by utilizing the Odyssey system and using Odyssey software or Image Studio software, you can easily place grids over data like this, quantify them, and then export them to a program of choice. Now let's move from phosphorylation to another example. Perhaps you're interested in protein trafficking. Now for this example, we're going to have two identical wells in, let's say, our 96-well plate. These wells have been treated identically, and the cells in these wells should be in identical state. And let's say we're interested in a receptor of choice. And our experimental question is, how much of my receptor is at the plasma membrane versus how much is internalized inside the cell? In one well, we'll do the In-Cell Western Assay that I've talked about so far. The second well, we'll put it through the variation called an On-Cell Western Assay. So for these cells, we've treated them, incubated them. Again, remove media. And then for the In-Cell Western Assay, as I've described, we'll permealize those cells. The On-Cell Western Assay will only differ in this one step in that it will not be permealized. What this means is when you add primary antibodies, and in this case I'm showing primary antibodies directed against an external epitope. For the In-Cell Western, these primary antibodies will recognize both our plasma membrane-associated receptors as well as internalized receptors. The On-Cell Western assay, since those were not permealized, primary antibodies will only be able to recognize plasma membrane-associated proteins. After we've added primary, we add secondary, and again, once we've added secondary, we'll read these plates on an Odyssey system, and then we can use the In-Cell Western assay cells to give us our total receptor content. So we'll get signals from plasma membrane-associated proteins as well as internalized receptors. The On-Cell Western assay, the signal we will see will only be those receptors at the plasma membrane. And so by comparing the In-Cell Western assay cells to the On-Cell Western cells, we can easily find out how much of my receptor is at the plasma membrane. Doing an assay like this comes in handy if you're looking at protein trafficking, receptor internalization after adding ligand, or receptor recycling after your receptors have been internalized. Other applications of the In-Cell Western assay are listed here. I don't have time to go through each one of these, but the In-Cell Western assay can be used for things like gene expression, viral titer load, apoptosis, screening RNAi libraries, and cell proliferation. I encourage you to visit licore.com/ICW. On our webpage, we have a lot of information, including protocols, workflows, application overview. There's a lot of helpful resources to help you get up and running with this assay in the lab. So let's talk about an example in research. And again, I'm going to talk about Alzheimer's research. Just to give a little background into Alzheimer's, very brief, but traditionally this disease is hallmarked by both A-beta-42 plaques and hyperphosphorylation of tau. Current patient treatments for Alzheimer's mainly focus on improving memory function and giving NMDA antagonists to decrease cytotoxicity. Now, since these don't address the cause of the disease, these treatments are not cures, but simply prolong the onset of disease. So in an effort to find a cure for Alzheimer's, typically groups have been looking at A-beta-42 plaques and hyperphosphorylation of tau. So with A-beta-42 plaques, looking at drugs that will block plaque formation. Now there's some evidence showing that even if you can block plaque formation, that dysfunction of tau in and of itself will be sufficient for disease. And so to block tau hyperphosphorylation, groups have been looking at targets such as blocking the tau kinase inhibitors, stabilizing microtubules, and inhibiting aggregation of tau. But what was asked was, well, perhaps we can increase chaperone expression. And by increasing chaperone expression, they can take care of the aberrant proteins and decrease, in this case, tau levels. Now, the proof of concept from this came from looking at galvanomycin. Galvanomycin in the early 90s was shown to inhibit HSP90. By inhibiting HSP90, groups saw an increase in chaperone expression and a converse decrease in overall tau expression. Now, although this could be great for treatment for Alzheimer's, the downside to using this drug was that it could not pass the blood-brain barrier. So what was asked by Dickey et al. In 2005 was, perhaps if we look at other HSP90 inhibitors and derivative of these inhibitors, select those inhibitors that could potentially cross the blood-brain barrier and ask, can they decrease tau levels? So what was done is they screened 10 HSP90 inhibitors. They treated cells for 48 hours with 1 micromolar of these inhibitors. This is what the data looked like. What we have here is tau levels shown on top, GAP-DH shown in green, which is our normalizing signal, and then HSP70 signals shown in the bottom. Now let's first focus on tau. We have our 10 inhibitors, A through J, and then on the far right we have vehicle-treated controls. Now again, the goal of these inhibitors is that they're going to inhibit HSP90, increase chaperone expression, and decrease tau levels. So when we compare this data to vehicle-treated controls, what you can see is these inhibitors varied with their ability to decrease tau levels. Some inhibitors, like inhibitor F, appeared to decrease tau greatly. Shown directly below is the corresponding Western blot. GAP-DH, again, is simply our normalizing signal. And then below is HSP70, which is a typical chaperone that the group expected to increase in concentration. So when we look at vehicle-treated controls shown on the far right, what you can see is all 10 inhibitors increased chaperone expression. Now again, by utilizing Image Studio software or Odyssey software, you can easily place grids on top of this type of data and then quantitate that data and export it to a program of choice. When we do that and plot the data, this is what you get. So what these are our tau signal and our GAP-DH signal. 10 inhibitors, A through J, and on the y-axis they're plotted against percentage of vehicle-treated controls. So gray is our GAP-DH signal. Again, since this is our normalizing signal, we would expect them to be roughly 100% of vehicle-treated controls. And then shown in black are our tau expression levels. So what was found is that some inhibitors, like those shown G through J, is that they did not significantly decrease tau levels. Whereas some inhibitors had a moderate decrease in tau expression, and one inhibitor in particular had a large decrease in tau expression levels. Again, remember the other question asked here was, how do these increase chaperone expression? Again, the data I'm showing here is for HSP70. And what was found by this group was, again, inhibitors A through J, plotted on the y-axis as a percentage of vehicle-treated controls. What was found is that all 10 inhibitors increased chaperone expression, but with varying degrees. So if you look at inhibitors I and J, they didn't have quite as large an increase in HSP70 expression. If you correlate that with decreases in tau levels, you'll find that these inhibitors also were ineffective at decreasing tau levels. So the key findings here for this group was that, yes, indeed, these HSP90 inhibitor derivatives can decrease tau levels with varying degree. They can also increase chaperone expression. I showed data for HSP70, but they also showed that there were increases in HSP27 and HSP40. But the key thing that was shown here, which was exciting, was that they have a potential to cross the blood-brain barrier. Because of that, these have a potential use as treatments and therapeutics in patients. But the key thing, to bring it back to the purpose of this talk, was that utilizing the In-Cell Western Assay, they can quickly screen compounds and screen replicates of compounds. And by doing that, they can find those compounds which give promising results, which they then can continue on and do more advanced biochemical analyses or potentially even animal experiments. In summary, the In-Cell Western Assay serves as an alternative to some of your typical Western blot experiments. Again, because we streamline the protocol and remove some of the steps in the protocol, it will save you time in the lab. Because we reduce the size of reagents used, you save costs in reagents. And ultimately, the data you get out of the assay will be of higher quality and more significance. And again, I urge you to visit licor.com/ICW, where we have a lot of protocols and information about this assay and how you can utilize it in the lab for the experiments you're doing today. And at this point, I'd be more than happy to answer any questions if anyone has any.